First of all, I would like to thank everyone who contributed to the grants that I received, and it's been a great motivation for me, as well as our group. Would also like to thank everyone in our group, Hague's Lab, as well as all our collaborators, and some other funds that we have, including the grants from the CJD Foundation Australia. So the basic question of our study is basically, what are the biochemical changes in the brain that may predispose individuals to memory and behavioral dysfunctions associated with prion diseases? As you all know, that our brain is made up of billions of interconnected neurons. And neurons are specialized cells. They can propagate and transmit electrical current. They can also talk to each other by releasing and receiving transmitters, which are chemicals. This network communication between neurons is, when it increases, is associated with how we learn new things and how we form new memories. And when it decreases, it's associated with when we forget things. When it becomes dysfunction, it is basically seen in neurological disorder where individuals would have dementia and behavior deficits. In prion diseases, all types of prion disease produce prions. The production of prions is associated with the degeneration of the neurons, and the degeneration of the neurons is associated with dementia and behavioral deficits. Memory and behavioral impairment in prion disease is associated with impairment of cerebral cortex activity. The impairment of cerebral cortex activity is usually detected during the prion disease by abnormal EEG or abnormal MRI. Note that the abnormal EEG is associated with abnormal activity of the neurons. During prion disease, there is a time where there is no symptoms of the disease, the asymptomatic stage, and then there's time where there's onset of clinical sign and symptomatic stage of the disease. The detection of the dysfunction of cerebral cortex activity is usually associated with, well, it's usually detected during the symptomatic stage of the disease. So what we are actually trying to find out in our study is what have actually changed biochemically during the asymptomatic stage of the disease that may predispose individuals or predispose brains to cerebral cortex dysfunction associated with prion disease. But the question is, can we model human cerebral cortex, and how do we know if an individual or cerebral cortex is likely going to develop the disease? So lucky enough for us, the advanced development in stem cell technology has allowed us to be able to grow mini brain in the lab, and by basically collecting skin cells from a real person, and then reset those cells to become master cells, and then from there we force the cells to form mini brain resembling the features of the cerebral cortex. So what is, and we call those mini brain cerebral organoids. So what is really important here is that these cerebral organoids retain the genetic information of the donors. So what we did, Dr. Zhou from Cleveland collected some skin cells from individuals who are carriers of E200K mutation, and in which the disease penetrance is pretty high. and we use those to grow cerebral organoids with E200K mutations. So now we have a model to study human cerebral cortex, and particularly in prion disease. As well, we know that our cerebral organoids would likely develop the disease given the high penetrance of the mutation. So that leads to the first aim of our study, is basically to investigate the inference of genetic mutation that predispose individuals to genetic prion disease with a readout on neuronal electrical signaling. So whenever I'm going to say from now on non-predisposed organoids, I'll refer to without the mutation, and predisposed organoids is for organoids with the mutation. So how do we know if our predisposed organoids are still at the asymptomatic stage of the disease? The resistance of E200K prion to a proteinase K digestion, which indicates the stage of the disease. PK is an enzyme that digests all proteins, and during prion disease, E200K prion species are highly PK sensitive at the asymptomatic stage of the disease, but when it gets to the symptomatic stage of the disease, they are mostly PK resistant. So this has been seen in terminal stage of human prion disease, where there's a substantial level of PK resistant prion at the terminal stage of the disease. In mouse model of the disease, where mouse carries the mutation, at the asymptomatic stage of the disease, there is almost no PK resistant prions. But at the symptomatic stage of the disease, you see substantial levels of PK resistant prions. In our predisposed organoids, up to 12 months, we have not seen any PK resistant prions. So that shows that we are still at the asymptomatic stage of the disease with our organoids. So we then decided to have a look at the neuronal activity in these organoids, the spontaneous activity, which is associated with memory and learning. And with our non-predisposed organoids, we put them on top of electrodes, microelectrodes. The black dots on the left are the microelectrodes, and the organoids are on top of it. And the box on the right shows the readout from those electrodes, the electrical signaling from the neurons in those in our organoids, and the green dots in each box indicates the electrical signaling measured or detected by each electrode. So as you can see, that there are some electrodes. That are next to each other, showing that they are a group of neurons interconnected and they talk to each other. And then with our predisposed organoids, they seem to have the same connection, neuronal talk, showing that the predisposed organoids have normal spontaneous activity as compared to the normal predisposed to the normal control. So although there is normal spontaneous activity in our predisposed organoid, they might have different ability to withstand stress, or in response to overactivation of neuronal activity. In this case, we decided to test the ability of these predisposed organoids by treating them with a glycine, which is a transmitter that stimulate the activity of the organoids, of the neurons. So overactivation of this activity of the neurons may lead to either way. Cell death would lead to cell death ultimately, but it can go either way. So either it will reduce and then cause cell death, or it would hyperactive or overactivate it and then leads to cell death. So we treated our organoids with higher dose of glycine and see what would happen. So what we found is with the non-predisposed organoids, they seem to get fatigue with higher concentration of glycine, whereas the predisposed organoids, they either increase or stay unaffected. So we then kind of conclude that non-predisposed organoid goes through different pathway to cell death compared to the predisposed organoid. So the predisposed organoids seem to get hyperactive and stay hyperactive until a stage where they might fatigue and die as a result of hyperactivity. So I usually think of it as the higher you climb, the harder you fall. So that's how I simply think of it. Our second aim of our study is to identify the changes of neuronal electrical signaling of cerebral organoids without the mutation that are induced by exposure to sporadic CJD extract. So our recent published paper showing that with this non-predisposed organoid exposure to infection, sporadic infection, they can develop the disease after 169 days. So we're thinking of recording the the neuronal activity of these organoids after infection and see what are neuronal activities that may lead to dysfunction that may lead to the infection and the disease later on in these organoids. So another future direction would be once we've identified some neuronal dysfunctions in these organoids, both the genetic and the sporadic, we're open for any compounds to test for potential therapeutic drugs. So in summary, I think I'm nearly running out of time, but again, once we've identified neuronal dysfunction in genetic mode of the disease as well as sporadic mode of the disease, we are hoping we may be able to find a way to rescue those dysfunctions in the future. So that's about it. Thank you.